Hello everyone, my name is Viviana Chavez, and today I'm going to be talking to you guys about canines as detectors and their disease management in agriculture. This is from my course Intro to Agriculture 10, and this is the fall of 2025. So, the introduction. Grapevine fields are vulnerable to diseases such as powdery mildew, and powdery mildew is a fungal disease that damages grapevine fields, affecting their yields and fruits. Traditional identification methods are usually visual symptoms and lab tests. However, this is a costly practice and often slow, and they usually miss early infections. Dogs can detect powdery mildew before symptoms even appear. Trained dogs have highly sensitive olfactory systems capable of detecting extremely low concentrations of chemical change compounds. Through research, we have found that dogs can be used for early detection of powdery mildew and in large scale plant disease management. This is important because canines trained to detect powdery mildew help to prevent loss of crops and chemical usage, ultimately affecting the public's health and improving by decreasing chemical usage. The background. Early detection can reduce crop loss, lower chemical usage, while improving sustainability and public health and the ecosystem. They can detect orders related to disease, including VOC chemicals that are released by cancer or plant pathogens. Canines can detect disease from breath, saliva, urine, and other samples. In other words, they don't require direct contact to detect a specific odor. They usually are used for biological scent detection because of their ability to detect without direct contact, which is used for crop disease management. Research gap. Many studies demonstrate that canines can detect human diseases, but few cover their ability to detect plant diseases. Agriculture detection relies on lab testing, or visual symptoms that appear late. Dogs have been used to detect plant diseases, but there is limited research done for grapevine detection and multiple disease detection. So I intend to study this by looking at the accuracy of trained canines at detecting fungal diseases such as powdery mildew and their VOCs compounds to human disease and compare it to human disease because it is still unknown through the lack of research. Research question. So, can trained dogs detect multiple types of fungal infections in grapevines before symptoms appear and with similar accuracy to human disease detection? My hypothesis is that if trained dogs can detect human disease with high accuracy, then they will identify multiple types of fungal infected grapevine leaves, similar to powdery mildew, before symptoms even appear. So my plan is to find sample plants and expose them to fungal diseases like powdery mildew, then collect 3 different stages of infection: the healthy, early infected, and the visible infected. Then I will select multiple trained detection dogs and train these dogs to detect infected leaves with a signal, usually sit. And then I will set up a controlled testing area with random leaf placements, present each sample to trained dogs in controlled environments, and record their detection. I will see if they find the correct detection, give me a false positive or a false negative. Then humans will walk around and do a visual inspection, and then we'll compare the results of the visual inspection and lab testing to the dog detection methods. And then we'll analyze whether dogs detected infections before symptoms appeared and if the results of canine detection and analyze the results of canine detection to human disease. I expect the dogs to correctly identify early stage infections through the VOC chemical compound, and visual detection will only identify disease after symptoms appear. Results may appear to show dogs are faster, more accurate, and more sensitive than other methods, supporting my hypothesis. In the chart below, you will see a sample test from a previous study where three dogs were trained to find the powdery mildew disease in grapevine fields, and they had a very high accuracy in finding them, and these are the results below based on the dog in the separate phases of testing. My project timeline will consist from January to May. In January, I will read the background on VOCs, disease detection, fungal infected plants. I will also gather supplies and possibly look into gathering some canines. Then I will talk with professionals about the project and gather advice for dogs available, and then form a final research question or hypothesis. Then in February, I will set up materials by gathering plant supplies for healthy and infected leaves. I will contact a local dog training facility to train the dogs for the project and learn to present samples safely, as well as prepare sample containers and controlled environment for testing. Then in March, I will work with trained dogs and run sample trials while recording responses. I will repeat the trial several times to gather sufficient data. In April, I want to analyze the results from the recorded responses into graphs and tables and compare the accuracy with research articles and look for patterns or similarities and identify limitations. In May, I will finalize my conclusion. Was my hypothesis correct or not? And then I will write my research paper draft and turn in my final paper. These are my references for this slideshow, and thank you for listening.